All right, so if you didn't quite understand the podcast over meiosis, here's another quick look at what's going on. Just a quick refresher on some vocab words. Remember, chromatin is our uncoiled DNA. It's going to coil tightly to make our chromatids and our visible chromosomes. Those sister chromatids are held together by a centromere. Our homologous chromosomes are chromosomes that possess the same genes, but not necessarily the same alleles. So you learned about alleles in 8th grade when you did genetics. This would be like a big T and a little t. So they would both have a T in that spot, but they're not necessarily the same size. Our tetrad is when our homologous chromosomes come together. A cell that is considered diploid has 2 sets of chromosomes in its cell. So here we have 2 red ones, 2 green ones, 2 yellows, and 2 blues. A haploid means that it only has 1 copy. So here we only have 1 set of each. Okay, so we're going to look through here really fast the different steps of meiosis. We're going to start off with the cell in interphase. It has a nucleus and it has its genetic material inside of the nucleus. So this is what it looks like during interphase. Remember, our nuclear membrane disappears and we start to see our chromatin coiling to eventually make our chromatids. So here we have a cell that has undergone interphase, it's undergone prophase, and now we're going to be moving on to metaphase. So here we see it finally in metaphase. Remember, we're lined up along the equator. Our homologous chromosomes have paired up. We're now having a tetrad. And now ideally, we're going to see crossing over at any of these spots where they combine. Because of the animation, we're not going to be able to see crossing over, but please realize that it will happen in these sections where the legs or the arms of our homologous chromosomes meet up. So as we move through anaphase, we see our splitting of our homologous chromosomes. We moved through telophase. Here we are in cytokinesis, where we have 2 cells that were each considered diploid. Now moving on into meiosis 2. So here we see prophase. They're going to line up in the middle during metaphase. Here we see anaphase. They're getting pulled apart. Telophase, they get completely pulled apart. We have the division of 2 cells. We're going to start to see the nuclear membrane show back up. Here we have our nuclear membrane. Each of my cells is haploid. They are different looking because of that crossing over. As they move through, they're going to start to go from their chromatid back into the chromatin form. And that is a quick overview of meiosis.